record on this computer. Okay, we are talking about immunity. We're talking about the immunity, the adaptive immunity that T cells and B cells give us. Now, T cells and B cells recognize foreign antigens. The first time they're exposed to a foreign antigen, they may take a while to spin up and prepare a response. But the second time that these T cells and B cells are exposed to the same antigen, they have a much quicker response. They've stored the memory of that foreign antigen in their cells. So while not every cell recognizes every antigen, there will be a handful of cells, T cells, and a handful of B cells called memory cells that recognize the antigen as foreign and will start reproducing quickly so they can destroy the cell with the antigen on it. This memory response allows us to very quickly eliminate a pathogen. In our first exposure to a pathogen, it may take 10 or 12 days before we recover from the infection, because it takes that long for the adaptive immune system to prepare and process a response. But if it's a pathogen we've already been exposed to, instead of 10 or 12 days, it may take 10 or 12 minutes or hours. And so the second exposure to a foreign antigen doesn't make us symptomatic. We aren't sick. We have a massive response of T cells and B cells, and we get rid of the pathogen before it harms us. That's what memory. Now, the other thing about our T cells and B cells is they are only going to attack the foreign substances. They recognize self. They recognize us as self, and so we don't attack ourselves. Only in an autoimmune disease do we actually attack our own cells. And then there, it's usually, even then, it's not systemic. It's usually just some location or some type of cells. So our T cells and B cells have become immunocompetent so that we don't go after a foreign cell. I mean, a healthy cell. Okay, now let's talk about active immunity. Active immunity occurs after we've been exposed to an antigen. Usually what happens is that once you're exposed to the foreign antigen, you develop an immunity to it. Now, the first time you're exposed to it, the first time you're exposed to a disease, you may become symptomatic. You may have been exposed to, chickenpox. Most people don't get chickenpox anymore, but some still do. But let's say you've been exposed to chickenpox for the very first time. You didn't have a shot, a vaccine to it, and you develop chickenpox. Well, now you have immunity. You have immunity to having it a second time. This is called active immunity. You have active immunity to a disease. And in this case, it's naturally acquired. If you developed a disease and got over it successfully, you're generally immune to it. People that would have developed smallpox 200 years ago would never get it again if they survived smallpox. When smallpox had a mortality rate, or has a mortality rate of 40%. But if the individual survived having smallpox, they were immune to it ever again. They had active immunity, and that was naturally acquired. Natural immunity occurs when you're exposed to the pathogen, and the antigens on the pathogen trigger your system to respond and produce T cells and B cells. So when you were exposed to the pathogen, you become ill, and then you recover. You have active immunity that is naturally acquired. It says here on the one bullet, natural active immunity results from exposure to an environmental antigen. You developed chickenpox because you were exposed to the virus. You developed smallpox, you were exposed to the virus, and you recovered. Artificial active immunity is a lot less dangerous and unpleasant. Artificial active immunity is when you are exposed to the pathogen, but the pathogen is either dead in the case of a bacteria or attenuated in the case of a virus, because viruses aren't alive. In other words, you're exposed to the antigen, but not the disease-causing properties. So you are exposed to a dead bacteria that has its antigens intact. So your body recognizes the antigen and mounts a response. You know, if you're exposed to we'll just say a dead virus. In other words, all you're getting is the protein shell. Remember, viruses have a shell around. The viruses are a DNA strand or an RNA strand surrounded by a coat of protein. The protein coat has the antigen sticking up on its surface. The same thing pretty much occurs in a bacteria. A bacteria is a living cell with a cell wall protecting the cell membrane, sort of like a plant. And then coming off the surface of the cell wall are these foreign antigens. Now, it's the antigens that are going to trigger the immune response, whether it is going to be the first time or the second time. So if you have natural immunity, it's because you were actually exposed to the pathogen, you became ill. Artificially acquired immunity, artificial active immunity occurs when you're given a vaccine. You're given a vaccine so that you are going to mount a response. You're going to have an immune response to the vaccine. You're not going to get sick. The vaccine doesn't make you sick. You get a flu shot. You don't get the flu from the flu shot. There's nothing in the flu shot that can give you the flu. What you're getting is exposure to the antigen. Your body doesn't know that the vaccine is not pathogenic. All it recognizes is there are foreign antigens on the coat of on the protein coat of the virus. And so it starts mounting a response. It produces lots of T cells and B cells. That's just what's known as the primary response. So artificial active immunity, you are deliberately exposed to a pathogen, a dead pathogen that can't make you sick, but it still has its intact antigens there, whether it's a protein, whether it's a bacteria or it's a virus. And we know viruses aren't alive, but we'll just call it dead for right now. So we have a dead bacteria or a dead virus. They're not pathogenic anymore because they're dead, but the protein, their coat with their antigens is intact, and it's the presence of the foreign antigen, the antigen that our immune system doesn't recognize, that triggers the immune response. So it's a whole lot better to get vaccinated than it is to get the illness. So active immunity. So how's it work? Well, when you become exposed to a foreign antigen, you have your T cells and your B cells are going to start responding. Now, if the T cells and B cells have seen it before, they're going to respond very quickly. But if they haven't responded, this is their first time, it will take a while for them to spin up the process. Now, what's going on here is that the B cells are going to make copies of themselves. Now, some of the B cells are going to attach themselves to the foreign antigen, or are going to have a receptor for that foreign antigen. And that B cell will say, hey, I match this antigen. So the B cell will say, okay, let's make copies of, this is a foreign antigen, let's make copies of ourselves so that we can find out and search out all the other foreign antigens just like this. So this becomes what we call an immunocompetent B cell. It recognizes the antigen as foreign. And so now we start growing more B cells that are going to identify that foreign antigen. So it's something like this. Here's the antigen up here, and here are the B cells, the B lymphocytes. Now there are receptor sites on these B cells that will actually, when a foreign antigen lands on the receptor on a B cell, it can trigger the B cells to start wanting to copy themselves, making clones of themselves. And so they will start making copies so that they can start catching, not catching, but so others of those foreign antigens can land on them. Now what will happen is these B cells that have now have a foreign antigen growing on them or attached to them. They're not harming the B cells, but they're on there. They're triggering the B cells to make what are known as plasma cells. Now the plasma cells make antibodies. So we can say that the B cells produce antibodies. Antibodies are proteins. The proteins glom onto the antigens. They can block the antigen from attacking a healthy cell. They can destroy the antigen. They can interfere with the antigens, the foreign antigen's ability to infect the cell in a variety of ways. And when it's the first exposure, it takes about 10 days to 2 weeks to produce enough antibodies to block the action of the antigen. And then you're exposed to a foreign virus, you get sick, and it's the flu, you didn't get a flu shot, you're exposed to it, it takes you 10 days to 2 weeks to get better. And then the whole rest of the winter, you never get the flu again, because even though you're exposed to it constantly, every time you go out in public or anywhere, you don't get the flu again, and it's not because you never encounter anyone with the flu virus. It's because now that your body has invested the time to make the antibodies for the pathogen, the flu pathogen, it stores that in memory. And the next time you get exposed to it, you don't have to wait 10 days to 2 weeks to feel better. You never become symptomatic. You can be exposed to the flu virus, and your adaptive immune system produces lots and lots of B cells, and they make a whole lot of antibody that gloms onto the foreign antigens, immobilizes it right away. You never become symptomatic. You're not even aware that you were exposed. The only way that we would know that you had been exposed to a pathogen would be measuring your antibody level in your plasma. That's the only way. And then after the exposure to the pathogen that you already have in memory, then it goes back into memory again and waits till the next time you're exposed, and the time after that, and time after that. So this memory lasts a lifetime. Our next response for an infection may not be till years later. You may be exposed to chickenpox once in your life and have what we call a primary response. And you go through all the symptoms, the headache, the fever, the nausea, the vomiting, the little lesions and scabs on your skin, and then you recover. And you never get chickenpox again, usually. You may have a dozen or 2 dozen, what we call a secondary response, which is the massive, overwhelming response of producing antibodies every time you're exposed to the virus, but you're not aware of it. So our B cells can produce antibodies years later, after the initial exposure. This is if, whether we are exposed to the foreign antigen through a vaccine or through natural exposure through the environment. So, okay. So how rapidly can we produce antibodies? Once we are in our secondary response, we are making 2,000 antibodies per second. So that's why in our secondary response, we aren't even aware that we've been exposed. The antibodies bind to the foreign antigens. They block their action or they mark them for destruction. And so we have memory cells and we have plasma cells. The B cells, the B cells that become sensitized to a foreign antigen, most are going to produce plasma cells to make antibodies. Some of the B cells are going to retain the memory of that foreign antigen so we can do the response again and again and again. So now, how many types of antibodies can we make? About 1 billion. We can generate about 1 billion different types of antibodies. So we can be exposed to about 1 billion different types of pathogens and respond. Because the older we get, the more antibodies we have available to us to attack foreign antigens. Because we're exposed to, as we age, we've gone through a period when we were younger where we were exposed to lots of things and we were symptomatic, as they say. But then, as we age, because the types of viruses and pathogens that are running around are probably still the same ones that were there or floating around when we were children, except for some of the newer ones like the COVIDs or all the coronaviruses. So now, this active immunity, we call it active humoral immunity. Again, it can be naturally acquired or artificially acquired. When we are artificially acquired, we're using the vaccine. Vaccines do not make you ill. Lots of people like to say, well, they got a flu shot and they got the flu from it. If your patient gets a flu shot and then develops the flu right after the shot, it's because they had already been exposed to the flu. It's not from the vaccine, because the vaccine doesn't contain anything in it to give you the flu. There's nothing there. So all that the vaccine does for you is trigger your B cells to start producing antibodies. They go through this, they produce the antibodies. They have what's known as a primary response. They produce the antibodies and then they retreat back into memory. And it takes about 2 weeks to get the antibody levels produced, and then they recede. And the next time you're exposed, you have a massive response in minutes to hours as opposed to days. So acquired active immunity. Flu shots, COVID shots. We've all gone through different types of vaccines. So we don't become ill. We're not supposed to become ill from the vaccine. Now that was active immunity, active humoral immunity. Remember the term humoral. Active humoral immunity. You generate your own B cells producing antibodies. You produce your own antibodies. Now, passive immunity is different. Now, active immunity, your immune system is actively generating antibodies and memory cells. Passive immunity is different. Passive immunity, the B cells are not challenged by the antigens. There is no immune response to the antigens. There is no memory in here. Passive immunity depends on borrowed antibodies. It's based on borrowed antibodies. Now, they're either naturally acquired, they've been, like when we are infants, we have antibodies that pass from the mother to the infant, through the placenta. That's naturally acquired. The colostrum that the baby feeds on prior to the production of milk in the mother's breast. The colostrum is very rich in antibodies. It's another way to deliver antibodies into the baby's body. That's naturally acquired passive immunity. The baby isn't making the antibodies, but the antibodies are being taken from the mother. That's passive, naturally acquired. Now, artificially acquired antibodies occurs when we inject a serum into the individual. Gamma globulin is a type of serum made that is rich in antibodies. In this case, where does the serum come from? Well, you take an individual who has been injected with the protein coat of a virus, and their immune system produces lots and lots of antibodies, or they're injected with a dead bacteria, and they produce lots and lots of antibodies to that dead bacteria. And then you draw off some of their plasma and filter it down and spin it down, and you get an antibody-rich substance that you can inject into somebody else. And those antibodies will only attack a specific pathogen. So what you do is you trigger the immune response in a healthy individual. They produce antibodies. You're not injecting them with the disease. You're injecting them with the dead cell or the attenuated virus. They produce an immune response. You filter off some of their antibodies and inject it into somebody else. It's artificially acquired. This is how the first disease was ever cured. First disease ever cured in the United States was diphtheria years ago, probably well over 100 years ago. Diphtheria is a bacterial infection. The way the serum was produced was to inject diphtheria. The diphtheria kills by generating a toxin which causes the trachea and the bronchi to swell shut, and your person dies from asphyxiation. It would kill lots. It seemed to affect children. It killed them the most. Anyway, New York Public Health Department developed a serum by injecting diphtheria into horses and then drawing off horse plasma and filtering it and condensing the antibodies into putting a lot of antibodies into a small unit of plasma, condensing it, if you will. And then that became the serum. It was the diphtheria serum that was injected into someone with diphtheria. And because it was borrowed antibodies, it worked. Didn't have to wait for the antibodies to kick in and be generated by your own immune system. The problem with that is, depending on your own development of your natural, of your immune system to kick in, your own natural immunity, you might die from the disease before you produce enough of your own antibodies to fight it. Some diseases are so virulent, so pathogenic, that they kill the host before the immune system can generate enough antibodies and T cells to fight it. And so, like Ebola virus, I mentioned this the other day, Ebola virus is so pathogenic that you didn't have time to, the body didn't have time to produce a response. Anyway, so a way around this for an extremely dangerous disease like diphtheria was to produce this serum, which was rich in antibodies, which would attack the bacteria that caused diphtheria and destroy it. And it worked. And that's sort of the term, that's how a serum would work. No antibiotics involved or anything like that. It was simply the antibodies. So, and of course, you know, breastfeeding is a great way to transfer antibodies from the mother through the milk to the infant, because at that point the baby's own immune system isn't functioning yet. During that first year of life is when our T cells and B cells are being formed and becoming immunocompetent. So here we see the different types of acquired immunity. This flowchart works pretty well for us. We have either naturally acquired or artificially acquired. Naturally acquired, we are going to, they're either going to be active or passive. Active, we're exposed to the pathogen, we have our own immune response. Passive, we get antibodies naturally from the mother. Artificially acquired immunity, active immunity from the vaccine. We get a flu shot. That's artificially acquired active immunity. Whenever it's active, it means that we have produced our own antibodies. Whenever it's passive, we have to depend on another source of antibodies. So whether it's naturally acquired, like breastfeeding, for passive immunity from breastfeeding or through the placenta. Here we also have artificially acquired immunity that's passive by getting an injection of a serum. We're using somebody else's antibodies to destroy the pathogen that we have. Active immunity generates a primary response. Active immunity will generate a primary response. It takes 10 days to get our antibodies up to speed. There is a 3 to 6 day lag after exposure to the antigen. We call it, that's the antigen challenge when it's recognized by the B cells. There's a 3 to 6 day lag in there when we're going to be symptomatic. But by day 10, we are starting to produce high levels of antibodies in our plasma so that we can start blocking and destroying the foreign pathogen. Then our antibody levels drop down. We've won the fight. The primary response occurs with active immunity. Because remember, active immunity, whether we're getting it naturally or artificially, depends on us producing our own antibodies. It takes time for the primary response to occur. It takes about a 14-day period. Now, of the different types of antibodies that we generate, the first antibodies that are going to show up at the site of an infection are called the IgM antibodies. They will appear first. The IgMs are going to start blocking the antigens. However, they don't generate a memory. After about 6 to 10 days, we start producing more and more of the second type of antibody called the IgG. And the IgG cells are slower because they're also producing memory cells. So IgM first, IgG second. These are the two types of antibodies that show up when we have an infection going on. The secondary response, we're dealing with active immunity, natural or artificially acquired. Active immunity always requires our own system with active immunity, it always requires our own system to produce the antibodies. And the primary response takes 2 weeks, 10 to 14 days. The secondary response, when we're exposed to the same foreign antigen again, takes hours, not days. Instead of peaking in 10 days, we peak in 2 days. And the level of antibody production after 2 days is probably double what it was the first time around. And the antibodies stay in the plasma for a much longer time. So while the first exposure gives us a response to get over the infection, the second exposure is like killing a fly with a sledgehammer and keep hitting the carcass with the sledgehammer to make sure that nothing comes back. And it looks, the curve, well, I'll get to that in a second. So what happens here, we produce a very large amount of in the secondary response, we produce a very large amount of the IgG antibodies. Remember, when primary response, we started out with IgMs, they come in, they lead the charge, they're going to start knocking down the antigen. They don't have any memory cells though. Then the IgGs gradually come in because they're producing memory cells. Well, in the secondary response, the memory cells are doing their job and having a massive influx of this IgG antibody, and they last a much longer time. Looks like this. This is the curve. See the blue curve on the left? Antibodies, primary exposure. First exposure here, primary exposure. You produce antibodies, and by day 10, 12, somewhere in there, you're getting lots of antibodies, and you recover. And you go off and you get exposed the second time. The first exposure, you probably were symptomatic of some, in some way, shape, or form. Now, the second response, though, is massive, and it only takes hours. Because look how many more antibodies you're producing here, and look how long how they tail out, so that you have protection to that antibody, to that antigen, much longer. So you don't become symptomatic at all. You're not even aware that you were exposed. Now, that only protects you for that particular antigen, because here comes another one right here, and it'll have the same response like you did over here. Antibody Y will give you the same response as antibody X did. It's two different pathogens. But then the second time you're exposed to this one, you'll get a curve like this. You'll get your secondary response is always huge. Now, antibodies, we call them immunoglobulins. How many of them are there? Well, there are 5 different types. They're produced by the B cells, by the plasma cells of the B cells, and there are 5 classes: IgD, IgM, IgA, IgG, and IgE. So they look like this. They look like a Y. They have a binding site on each end of the Y arm, and they have a location where complement, remember complement from the non-specific, where complement can bind on there. But your typical antibody is going to look like the letter Y. So these 5 classes of antibodies, IgM comes out first when you're exposed to a pathogen. IgG comes out second. That's fourth one down there. IgG is the antibody that will gradually start producing the memory cells. IgA is the antibody that's found in our fluids, saliva, mucus, sweat, gastric fluid, in our tears to protect us. IgG functions as an antigen receptor, and IgE down at the bottom is what we use to respond to allergic reactions. So when we're exposed, a person's having an allergic response to something, they're producing lots of IgE antibodies to try and block the antigen. It doesn't matter if it's pollen. Pollen has antigens too. It doesn't matter if it's bee venom or peanut oil. There are going to be foreign antigens on the surfaces of those structures, and that's where in that case, the IgE antibodies are going to be trying to fight back for you. So IgM is first, and then IgG comes in with a much larger response. And again, this is what our primary and secondary responses look like. Now, here we see it broken out. IgM comes out first and then tails off. IgG comes out slower, but gives you a much longer and more powerful response. Even our secondary response right here, with our exposure to secondary response, the IgG, which is based on the memory cells, gives you this much larger response that lasts longer. IgM comes out too, but because that's what it does. But in this case, it's not first, it's second. And it helps, but the IgG is doing a lot of the work at this point. Antibodies don't kill the antigen. They block it. They block its action and they tag it for destruction. They form what's known as an antigen-antibody complex, and they have 5 different mechanisms actually 4 different mechanisms to eliminate the antigen. They're neutralization, agglutination, precipitation, and complement fixation. So when they're neutralized, they simply prevent the antigen from getting into the target cell. They block the receptor site on the target cell. They block the binding site on the antigen. It can't get there. Agglutination and precipitation, the antigens that bind to the antibodies, and they clump together, and they can no longer land on the target cells. Complement is activated. The binding site on the antibody for complement is activated, and complement then can start punching holes in the bacteria and prevent it from landing on the target. Opsonization actually connects this is a process where the antibody ties the foreign antigen to a macrophage, and the macrophage then can destroy the foreign antigen in here. And we can trigger inflammation. The IgEs, and this is where we have that response with allergies. The IgEs will trigger a histamine release. When the IgEs become activated, see, histamine is not our enemy when it comes to allergens, even though we think it is, because we have a runny nose and we have watery eyes, on a minor case like pollen, or a major case like bee venom. Histamine is our body's way of trying to fight back against the foreign antigen. And so this is all part of the inflammatory response. And here are all the ways that our antibodies try to protect us through neutralization by blocking the site, agglutination, precipitation, complement fixation to punch holes in the bacteria or virus coat so it can't work. We release histamines to try and flush away the irritant. So all these ways working here. We don't actively destroy the antigen with the antibody. So So we talked about allergies and stuff like that already. Okay, let's move on. Now, cell-mediated immunity. Antibodies don't work against intracellular antigens, antigens that have penetrated the cell. So that's where the T cells come in. T cells allow cellular immunity to take place. Now, there are two types of T cells. We have what are called the CD4 T cells, which are helper cells, and the CD8 T cells. These are the cytotoxic T cells. These are the only cells that destroy foreign antigens. They can destroy cells that are infected. These are the only cells that destroy infected cells. They also have memory cells. They have suppressor cells too. So here we see a lymphocyte from the red bone marrow. It can either become a CD4 or a CD8 in the thymus. If it's a CD8, it will produce cytotoxic T cells. If it is a CD4, it will produce helper T cells. We need all of these. T cells will only work if they recognize the antigen that's brought to the surface of the infected cell. So a cell that's infected will alert the immune system by bringing fragments of the foreign antigen to the surface of its cell membrane. That tells the T cell, hey, that cell is infected. We need to go over and kill it. You know, we're not going to worry about the antigen. We're going to kill the cell. So cells that become infected, viruses, bacteria, any kind of parasite, cancer cells, cells that are abnormal in any way. Transplanted tissue cells are recognized as foreign also. And our T cells, the CD8 T cells will go after them. So now there are two types of proteins on the surface of all of our cells. And these are self-identifiers. They're a type of glycocalyx. They are not recognized as antigenic. They're not recognized as foreign. They're recognized as us. And the two types of proteins are called MHC proteins, class 1 and class 2. Class 1 MHC proteins are found on all of our cells. Class 2 are only found on certain cells during the immune response. MHC proteins, they are unique in each of us. What they will do, though, is they are formed inside the cell, and they migrate to the surface of a healthy cell all the time and bring up fragments of proteins from inside the cell. So they're coming up from the inside of the cell constantly. If they pick up foreign fragments, that will alert the T cells. If the cell is infected, these MHC proteins will bring up fragments of the antigens to the surface, and the T cells will say, oh, infection, let's go do something about it. As long as the fragments that are coming up, as long as the proteins, which are always breaking down, as long as the protein fragments are us, it's fine. But if we're bringing up foreign fragments, then we recognize it as something that needs to be taken care of. So something like this. Here's our antigen right here inside the cell. Fragments of that antigen end up attached to the MHC class 1 protein, which brings it to the surface of the cell membrane. And now a T cell can recognize it as a foreign substance. Now, class 2 is a little different. In the class 2, we are going to engulf a bacteria, the cell engulfs a bacteria, and we discover that there's an antigen on the bacteria, and that the class 2 will bring the antigen to the surface of the cell. It's after we've already, we're processing the bacteria and we bring it to the surface. Now, who gets involved with the class 2 cells? Well, class 1 MHCs are always recognized by the CD8s. Remember, the CD8s are the cytotoxic T cells. Always recognize these. So the class 1 MHC proteins always trigger the cytotoxic T cells. The primary response for a T cell takes about a week. Then the T cells break down. But the memory T cells are there to respond, and not in days, but in hours. So if you're exposed to the same pathogen again, you'll get a massive response again. The CD4 cells make the memory cells. The CD4 cells produce memory cells and allow the production of the cytotoxic T cells. Without the CD4s, we don't get an immune response. We don't get B cells and we don't get T cells. We've got to have the CD4s. And we also produce helper cells that tell the B cells to produce antibodies and to divide very quickly. So bullet here, bottom line, T cells that contain the CD8 proteins interact with, the CD8 T cells interact with class 1, the CD4s interact with class 2. Got to have the CD4s. Without CD4s, you don't get the immune response. You don't get T cells, you don't get B cells. And these cytotoxic T cells are the only cells that can attack and kill other cells. They go through our body looking for things to kill. They like to hang out in the lymph nodes. Where else are we going to get to filter everything? And the natural killer cells are a type of these cytotoxic T cells. So, and this is what, here we see one going after a cancer cell right there. So, memory T cells don't get involved in the first encounter. The CD4 cells are going to produce memory cells too, but the memory cells will kick in for the second exposure, and we'll have a response very similar to what the B cells are producing for us. Okay, and that is the end of the adaptive portion of the immune system.